All right, I don't want to have everyone wait too long, so I'm going to go ahead and introduce our speaker tonight. And I've actually known him since 2011, I think. We rotated through theriogenology together in vet school, but he's actually from Australia. And since then, he's done quite a bit. He's a bit unique at Cornell in the sense that he's double boarded, which means that he has two specialties. So he's both a theriogenologist, which is reproduction in animals, and then he's also a large animal internal medicine specialist. And he has a Ph.D. because I suppose he was bored at some point. And so he's highly qualified and has a lot of pretty cool, diverse interests. And he's one of his many interests, so obviously, is genetics. He's, of course, involved in that through both of his specialties. And he's going to touch on, I think, some of the breeding side as well as some of the health implications. So really happy to have him. And thank you, Dr. Donnelly.
Thank you, Lindsay, for the introduction. For the talk, I'm going to use the laser pointer if I have things that I'm going to draw your attention to as we go along. I have nothing to disclose. I do genetics research. I've been involved with a lot of projects that have discovered different variants that have disease-bearing sort of nature, but I'm not involved with any genetic testing companies. So I kind of wanted to get us all on the same page for some genetic literacy. So what are the terminology that we use and how we sort of talk to one another about the genetic basis of health and disease. And so at the very sort of beginning of that, all of our mammalian species have chromosomes. And in horses, we have 32 pairs of chromosomes. And that's where the name of the talk comes from, 32NU. It's a bit of a spin-off of the 23andMe, which would be the number of paired chromosomes that we have. With the chromosomes, we have what we call autosomes, and then we have what we call sex chromosomes. So in horses, we have 31 autosome pairs and 1 pair of sex chromosomes. Our autosomes are very creatively named 1 through 31. And then for sex chromosomes, we either have XX if the animals are female or XY. And collectively, we like to say chromosomes wear genes as their outfit. And so each chromosome encodes, several thousand and collectively around 20,000 coding genes. And when we talk about coding genes, they're genes that produce a protein, and proteins are the things that sort of make our body function. If we look at a deeper level, what chromosomes are highly concentrated DNA molecules. And so DNA, we think of it as this sort of double helix structure as the sort of classic thing, but in reality, it's very tightly bound and condensed into our chromosome. And so sort of to reiterate, we have 2 chromosomes, and on those chromosomes are genes. And so for every individual, we have 1 version of the gene on 1 chromosome and 1 version of the gene on the other chromosome. And that's what we call an allele. If we have the same alleles on both chromosomes, that's what we would call homozygous. And if we have different alleles, that's when we have a heterozygote condition. If we look sort of broadly at the field of genetics, it's broken down into some disciplines. And so the first one is population genetics. And I think this is probably what a lot of people think about genetics. And population genetics is really how an individual relates to other individuals within its breed. And then, if we look more widely, how each breed interacts with one another. And so this is a highly statistical form of genetics. And we have an example here in this circus plot dendrogram that maps out the genetic distances of certain breeds from one another. So we can see Shetland ponies and Icelandic horses segregate together as a more closely related breed than do Belgian draft horses to Shetland ponies. Most of what we're going to spend time talking about today are molecular genetics. And molecular genetics really refers to looking at those nucleotide sequences of the DNA and how they might change in health and disease. And so we have a number of ways that they might change. Some simple ones would be substitutions. And so in this example, where we have our C, it's mutated and becomes a T. And insertion is where we have extra nucleotides. And so in this example, we have the motif here of a TAT being inserted between this G and this C. And then in the case of a deletion, we remove those nucleotides. And so this CA between the two Gs is missing. These may cause disease. They may cause no disease. And this is a sort of natural part of variation in everyone's genome. An important and sometimes overlooked discipline of genetics, and we won't go into this a lot today, but is cytogenetics. And cytogenetics really is the study of chromosomes at their sort of macro level. And so we're studying the structure and function of our chromosomes. So again, in our horses, those 31 autosomes, and then our 2 sex chromosomes. And the last area, and is kind of where the field of genetics has gone into, is what we call genomics. And genomics really, all it means is we're studying all the genes of an organism at the same time. And so again, we see this progression here. We have our chromosomes. So on one side, if we're studying a single gene, this would be more what we would call genetics in its classical form. Or if we go to the other way, where we're looking at all the genes on all of the chromosomes collectively within an individual or across populations, this is what we would call genomics. So horses have a really rich history of using genetic tests as a way of promoting equine health. And so the first published genetic test was for a disease called hypokalemic periodic paralysis. And if that's a bit of a tongue twister, you're amongst good company. This was a condition mostly characterized in quarter horses and was discovered in the early 1990s. So I'm sure if people have halter type quarter horses, they would be aware of this condition. Colors were the next thing that came along. And so the causative variant, and I use variant here instead of mutation, they're sort of interchangeable, but a variant would be how we would describe them now, was for chestnut. And this was published in 1996. Sort of fast forward from there, we've gone from that sort of genetic age of looking at single genes, very laborious, time-consuming work that was a sort of slow grind to put together the phenotypes, or what the animal expresses physically, or in medical terms, with the genotypes, so the genes that were involved. And so the thing that really has changed for equine genetic health was the publishing of the equine genome. So the equine genome was first published in 2009, which was only 6 years after the human genome. And Cornell was very much involved with this. And the first equine sequence was based off of Twilight, who lived at McConville and up until recently, and is, sort of our superstar queen of genetics. So before 2009, we knew about 14 color traits and about 10 health traits that a lot of work had gone into over many decades to sort of produce that number. Now that we have a reference genome, so we know where those A's and C's and T's and G's are meant to be, we've seen this rapid accumulation of knowledge around health conditions in horses and also traits that cause color. And so post publishing of the genome, we now know about 44 additional color variants and 24 additional health variants. Some of these are a little bit overlap in that they may be the same phenotype. So it might be that we know more about a bay variant or a chestnut variant that is specific to a breed, but it's still sort of included in those numbers. And color traits tend to predominate in discovery because they're very readily phenotyped. Like you can go out and see that a horse has a particular color and put a group of horses together, analyze them, and then find the variant. After the initial publication of the genome, there was an update called EQUAB3, and that came out in 2018. And we can expect some more discoveries from that because it has added certain sort of advantages, including a Y chromosome to the assembly. So when we talk about genetic testing, there are different types of tests that we might be able to run. And in horses, there are sort of two main ways. And the first one would be single trait testing. And so this is typically breed specific. If there is a known condition or color variant in a breed, so we test an animal for a single gene, and we do this, either by typically by a technique called PCR. And so the example here is Connemara hoof wall separation disease. This is autosomal recessive, and we'll go through some inheritance patterns very soon. Disease that only affects Connemara horses, and this variant was discovered in around 2012. And so if you have a Connemara horse, and now with the Connemara breeders, hoof wall separation testing is a part of the breeding program. And so we can see that we would get information about that one gene. The alternative is panel testing. And so when you submit your sample, you'll get back a panel of different genes. So each gene individually tested to see if you have the relevant disease-bearing variant, or if the animal has the reference allele. And so an example of that would be the AQHA 5 panel. We now say the 5 panel plus because it includes more than just the original 5 diseases that were mandated by the AQHA. Importantly, ones that have been added recently would be the MYH1 or the myosin heavy chain 1, which has been associated with immune-mediated myositis and also with some non-exertional rhabdomyolysis. There are some other ways that we can do genetic testing. One of those would be what we call a SNP array. And SNP arrays essentially are panels of single nucleotide polymorphisms or variations within the genome spread out through all those chromosomes, and they're based on a number of different breeds. So it's sort of breed irrespective and spans across different areas of the chromosome. It doesn't analyze every single nucleotide, and so it sort of gives you regions, and it's not necessarily the current way that we test for diseases, but is sort of more of an exploratory method. SNP arrays, this is how, if you have dogs and you've done some genetic testing in them, this is typically how those tests are done, but are yet to sort of really be adopted in horses. The level up from that is what we would call whole genome sequence. And this is where we detect every nucleotide in the animal's individual genome. This is really useful and powerful if we have what we call a de novo trait, and that is something that arises in a particular individual. And the example that I have here, this Palomino stallion, this was the sire and the dam, neither of which had a white patterning variant, and they produced a white pattern foal. When they tested this foal for all known white pattern variants, he was negative. And so it suggested that he had a new variant, and this was the first documentation of what we call splash white 6. Now, it occurred in a gene that has been published before for causing white pattern variants, but the specific type of mutation that this animal had was yet to be discovered, and it was only able to be discovered through doing whole genome sequencing. As whole genome sequencing becomes more cost effective, it's probably something that we will be doing more often for patient horses as they do in humans. And the sort of the threshold in humans has always been the $1,000 genome, and we're right around that $1,000 a genome right now. I think we need to come down a little bit more for our horse patients. But I would say within the next few years, we'll probably be pretty close to being in the hundreds of dollars per genome. And the last one, and again, a very powerful technique, and a sort of classic one in genetics, is the karyotype. And this is particularly useful in horses because horses are one of the few species outside of humans that have a frequency of aneuploidies that survive into adulthood. And what aneuploidies mean are aberrations in the number of chromosomes. So a common example would be trisomies in humans. The sort of classic example would be Down syndrome. In horses, we often see extra sex chromosomes, so an extra X or an extra Y, or we have only an X. And these are things that can only be diagnosed through using a karyotype. And so that becomes a useful technique, but we won't necessarily talk too much more about that. I'm going to move on to how variants are inherited. And we're going to talk about this in the sort of classic Mendelian fashion first, and then go into a little bit more about disease risk. So the first one would be recessive traits. Now, the sort of classic feature of a recessive trait is that it is equally carried amongst males and females, and that only animals with two of those variant alleles, or what we would call homozygous, would be expressing the phenotype for the disease condition. And so we see over here, this is a way of doing a pedigree in sort of science language. So by convention, males are square and females are circles. And so we see equal carrier state amongst males and females. And then when we have two carriers that produce offspring, we have the potential to produce a homozygous normal, or homozygous reference, we would call this, heterozygous in both male and female, and then the potential to cause a homozygous recessive or homozygous alternate. So if we look at horse disease specifically, and this is a relatively recent one, and that's why I wanted to highlight it. It's called equine familial isolated hypoparathyroidism. Has a very, very long name for a disease that is of pretty high consequence, especially to our thoroughbred breeders. And it affects a gene called RAPGA5. And this is a recessive trait. And so we either have animals that don't have the variant allele, so are normal and non-carriers. And then we have animals that do have the variant allele, they're normal and the carriers, or if they have both variants, then they would become affected. And this is the sort of disease-bearing part of the inheritance. So you need to have two copies of those alleles that are diseased to produce an affected animal. Now, it's at a relatively low rate amongst thoroughbreds. It hasn't been identified in any other breeds. Has a carrier frequency about 1.5%. But that means in the United States, it's estimated that about 600 foals per year are produced that are carriers. And the reason why it's really important to know about carriers is that this disease is universally fatal in the neonatal period. These foals are not able to manage their own calcium levels, and so do not make it into adulthood. And so in the thoroughbred population of, say, racehorses, you're never going to see an animal that has two copies of this gene because they don't survive. But you may see animals that have one copy. And if we were to get into a situation where, say, a stallion that became very popular was a carrier, this would be important to know because it may increase this carrier frequency. The carrier frequency, as far as we know, for the last about 30 plus years has remained fairly constant. And so it's sort of at a background level, which is a good thing. The next mode of inheritance is what we call dominant. And again, it's equally affected for males and females. What's different about dominant inheritance is that it does not have to be homozygous alternate to produce the phenotype. So that means you only need one copy of the variant allele, so the one that causes disease, to see a phenotypic effect. And so this can be on two ways. So we can have an individual that has two disease alleles, so is dominant on both of those of genes that they inherit from, one from mom, one from dad. Or they can be heterozygous on either allele. And in both of these situations, we will see a phenotype effect in a dominantly inherited disease. The only time where we will not see an effect is if the individual does not inherit one of the dominant alleles. And so when you have one parent that is homozygous dominant, you have 100% chance of producing heterozygous, whereas a heterozygous dominant, it's around a 50% chance. of producing a heterozygous dominant. So the classic dominant disease in horses is malignant hypothermia, and this was one of the earlier genetic diseases that was discovered in horses, and it affects this gene in the ryanodine receptor. It's the same thing that happens in other species with malignant hypothermia, like pigs, and people also get this. And so when we do our test and we get it back, we're either going to have an animal that does not have any of the variant alleles, or they will either be heterozygous or homozygous, but in both cases, they will be affected. The trick with malignant hypothermia, though, is that it's a disease plus exposure risk. So you need both to have the genetic variant plus the risk factor, so normally a medication, to produce malignant hypothermia. What's interesting about malignant hypothermia and what makes doing genetic testing important beyond this sort of one pathway is that this gene has a modifying effect on other genetic diseases. And so this one in particular is PSSM1, so polysaccharide storage myopathy 1. Both of these are relatively common in quarter horse type breeds. And if you have both the malignant hypothermia variant plus the PSSM1 variant, it can make PSSM1 worse. And so that's where a dominant trait is having a sort of confounding effect or compounding effect, sorry, on a different genetic disease. One that is often a little bit harder to wrap your head around is what we call an incomplete dominant trait. Now, it is inherited in the same way that a dominant trait is, in that you only need to have a single copy of the variant allele to have a phenotypic effect. What's different is that we normally have two different phenotype levels. And so if you have a homozygous dominant animal, we'll have a dominant phenotype that will be different from a heterozygous animal that will have an intermediate phenotype. And again, our homozygous reference, so that one without the variant allele that is acting in a dominant inheritance pattern, will not show the disease. Again, this is also sort of equally distributed between males and females. And so probably one of the best examples in our horses is the leopard complex spotting. So the TRPM1 gene that is sometimes called LP. And if we only have one copy of LP, it causes two, or at least two different coat color phenotypes. So the blanket phenotype and then our sort of lovely leopard phenotype. And that's what we call the intermediate phenotype, when we just have one copy. When we have two copies, we see different coat color phenotypes. So we have the blanket or the few spot Appaloosa. And in this sort of double dominant situation, we also have the addition of a different phenotype, which is the congenital stationary night blindness, or sometimes just called night blindness. I'm going to talk a little bit more about that in a minute. And so we can see that it's the one gene, but depending on how many copies of that gene will dictate what kind of phenotype you have in that horse. The last mode of inheritance that I'll talk about is what we call sex-linked, and this is when a variant occurs on either the X or the Y chromosome. Much more commonly is this an X chromosome disease. The Y chromosome, depending on the species, isn't very big, and so doesn't really have a lot of room for many genes. And so classically, this is a situation of where you have mostly males affected if it's an X chromosome condition, and you have females that are carriers only, but are never able to, or very, very rarely have the disease or are affected by the disease. And that's because females inherit two X chromosomes, and a lot of the time one is sufficient to make up for the deficit of a variant. So if they're a carrier, they have their other X chromosome in what we call X inactivation. And so in this example here, we see a carrier female and an affected male. in this context, again, this affected male is going to produce carrier females. But if this carrier female were to be bred to an unaffected male, we could see a situation where we have an affected male. And so the most recognized X-linked trait in horses is what we call androgen insensitivity syndrome. And so this is a situation of sex reversal. So we have a genetically male but phenotypically female individual. And so they have the normal number of chromosomes, they have the typical male XY chromosomes, and they have what we call SRY positive, which is the sex determining region of the Y chromosome. So in development, that is what sort of dictates developing into a male, but they lack the androgen receptor, and the androgen receptor is encoded on the X chromosome, which is our, female-bearing chromosome. And so there are a number of different mutations that have occurred in horses, and so this is a trait that has sort of been recognized in a various number of breeds. But once we have a mutation there, it acts in a sex-linked, and in this case specifically X-linked fashion, so that males are affected or normal and very uncommonly a carrier. So they're either affected and inherently a carrier, or they will be normal. Females can be carriers and or normal, but they would not be affected. It's a very, very rare situation where you would have a homozygous for an X-bearing trait. So sort of different to our other modes of inheritance in that there is a sex bias. So typically it's going to be males if it's an X-linked trait. There are some other factors that go into inheritance, and then what we see on the phenotype level. So what that gene tells the protein to make, and then what that protein looks like to us. And so one of those things that can be a little bit challenging sometimes when we look across breeds is that we have what we call genetic heterogeneity. And a good example of that is stationary night blindness. So we have two breeds here. We have our Tennessee walking horse and we have our Appaloosa. Appaloosas have been for a long time recognized to have congenital stationary night blindness. And so if you take two horses that have the potential for congenital stationary night blindness, and you do what we call an electroretinogram. Over this side, we have our normal electroretinogram, so our OD and our OS are our right and our left eye. And in a normal animal, when you give them a pulse of light, we have these two peaks, our A and our B wave, that tell us that the retina is responding to that light in dark adapted conditions. In an abnormal animal, it's kind of a flat line. What we see is that in both Tennessee walking horses, standardbreds, and Appaloosas, we can see the same phenotype. But what's different is that in our Tennessee walking horses and standardbreds, it's caused by a gene called GRM6. And so we now call this CSNB2, or congenital stationary night blindness 2. And then we have on our Appaloosa side, TRPM1, which is our original congenital stationary night blindness or congenital stationary night blindness 1. So it's the same phenotype, the same trait, but two different genes are responsible for it. And that's what we call genetic heterogeneity. So both horses, both with the same disease, but different genes. Now, both of these genes work in the same pathway, so it makes a lot of sense that they would have the same phenotype. But it's just to sort of say that you can have the same genetic disease, but different genes can cause them. Sort of along with that, we can have what we call phenocopies. And phenocopies are probably the more troublesome sometimes, because these are the ones that often get a little bit confusing when we're doing genetic testing. And probably the poster child for that is polysaccharide storage myopathy, or PSSM, which I'm sure a lot of you, if you have quarter horses or draft horse type horses, will be aware of. So when we have a horse that is PSSM1, it has the specific mutation plus the phenotype. So the phenotype is specifically increased glycogen deposition in muscle that's irregular. And that's the polysaccharide storage part. And that's only diagnosed by biopsy. And those two things were put together so that now when we test a horse for PSSM1, that mutation, we can be really confident that that's what's causing our myopathy. There are other horses, though, that get increased muscle glycogen or erratic storage of that muscle glycogen. That looks almost exactly the same as PSSM1. And so this is what we call a phenocopy, because these horses do not have the same genetic variant. And so we kind of lump all those horses in together as PSSM2. Now, there's no genetic basis for PSSM2 currently. There probably, maybe something discovered in the future, but as it currently stands, we only have a test for PSSM1, not PSSM2. And so this is what we would call a phenocopy. So same phenotype, but one is genetic and one is, potentially genetic or has some other environmental component, say the diet of the horse. Another sort of, or in the last kind of layer when we're looking at our genetic testing is what we call pleiotropy. And that's where a single gene may cause two or more phenotypes. And so if we think back to what we're talking about with our Tennessee Walker and our Appaloosa, they were the same phenotype caused by two different genes. This is kind of the opposite. This is two phenotypes caused by the same gene. And the best example of this is the silver gene. And so this is a gene called PMEL or PMEL, and it's a coat color producing gene. So in black and bay horses, it will turn that sort of black mane into a sort of flaxen color. So black horses become silver, bay horses become chocolate. It's not necessarily readily identifiable in chestnut horses. They still look chestnut, but it acts in a dominant fashion. It also causes an ocular phenotype. And when it causes an ocular phenotype, it acts as an incomplete dominant trait. And so it causes a disease called multiple ocular anomalies that causes these cysts in the back of the eye or holes in the iris called coloboma. And if you have just one copy of that, you have a sort of mild phenotype. You might have some cysts and that's about it. Or if you have two copies of the gene, it will cause a more severe phenotype. So I'm going to move on now, sort of just the last few little bits about genetic risk, and then also a bit about parentage and some of our future things. Something that's kind of emerging, and is probably the area that I'm most interested and active in, is this idea of using genetics as a way of improving health traits that aren't inherited in a strictly Mendelian fashion. And so that means that they may produce what we call genetic risk. And a really good example of that is the DDB2 variant that is fairly common in our Haflinger horses and in Belgian draft horses and a couple other breeds. And it is a risk factor for producing ocular squamous cell carcinoma. And so it doesn't mean that other horses don't get ocular squamous cell carcinoma. For sure they do. If you have a paint horse or a Clydesdale, all these horses that sort of lack pigment around their eyes, these are all horses that will also get this condition. But these horses are particularly susceptible to it if they have this variant and if they have it in homozygous form. So if they have two copies of that variant gene, they have an increased susceptibility to UV damage, and so therefore an increased susceptibility to squamous cell carcinoma. And if you have a Haflinger and that Haflinger has two variant DDB2s, it's almost 6 times a greater risk of developing ocular squamous cell carcinoma. And the Belgian draft, it's about 4 times. This variant explains about 80% of cases of this cancer in these horses. And so it's a really important one to test for if you have these breeds, because it kind of will help you manage that horse. It might be that you have a more night turnout predominant, or you're making sure you're using UV protection, those sorts of things. Something that I think a lot of horse owners are interested in are performance traits. And to be very honest, in the genetics realm, we know a little bit about some, but not a lot about a lot. And that is probably because there are things that aren't that easy to phenotype. Performance is particularly hard to phenotype. If we're trying to track times or jumping height or something like that, it's quite hard. Something that's relatively easy to phenotype, though, is how a horse moves. And so this is why our gait ability has a genetic test. And so it's what we call a permissive to gait. So this work that led to this was actually quite groundbreaking because it was sort of stuff that was done in horses, and they recapitulated it in a mouse, and then they were able to show very, conclusively that this single nucleotide change in the DMRT3 gene affected how the spinal cord works, and then the ability of horses like Icelandic horses to express all of the different gaits. So horses with the trait are much more likely to be able to pace, trot, colt, all of the good things. So like the 5 gaits, more so than other horses. And if we think about horses in our area, that is probably pretty common with the standardbred. Most standardbreds are fixed for this trait, and so that's why they're able to perform, in pace races more easily. And so how the test comes back to you is that you'll either have this AA or 2 alternate alleles, CA, so a heterozygous, so 1 alternate allele, or CC is the reference allele, so a homozygous wild type. And while these animals are able to perform these gaits, it's much, much easier for animals that are homozygous for the variant. We're just going to finish up in the last couple minutes here. Something that is offered by a lot of testing laboratories is the equine parentage testing. In some other species, we use what we call SNP-based parentage. In horses, we still use short tandem repeat. So these are areas within the genome that have repeating sequences. So either, an A and G, AT, that are flanked by more variable sequences, and they're sort of pretty conserved across horses. And essentially what happens is we match the number of repeats that are in the foal with the number of repeats in the dam and then potential sires. What the sort of take home for this is that they need to use about 15 markers, and one of those markers says if the foal is male or female. It is much, much easier to exclude an animal as a parent than it is to include them. It's more hard to prove that something is a parent than it is not. So that's kind of the take home on parentage testing. So even though it says 99% accurate, if you include both potential parents in the testing, part of the quality assurance for this is that they want to provide sort of 100% accuracy. And so it comes with that disclosure. And then if you only provide one parent and then no other potential parents, it becomes a little bit harder to identify. Something that I think is becoming of interest to horse owners because they see it on the dog genetic side, and that's breed composition testing. And this is a fairly complicated area of the field at the moment, and I'll very briefly sort of show you why. So originally, Dr. Peterson back in 2013 published this paper using SNP, so that panel that has little markers all across the genome, to see where horses stacked up based on breeds. So there's a whole group of different breeds here, and this is what we call a dendrogram, so dendro like tree. And you'll see that they branch off here, and that horses are sort of genetically distanced from one another based on the number of variants. There aren't that many branches, and that means that some horses are going to get grouped with one another, okay? If we look at it on that whole genome level, so knowing every variant that's in the genome, this is some work that Al Abbott done, and this is each dot represents one horse, and each dot represents all the variation in that horse's genome. And so at our extreme ends here, we have our thoroughbred horses in the sort of green, and then our quarter horses in the purple. And this PC1 where it says 5.91%, that means sort of the difference that explains between essentially quarter horses and thoroughbreds is only about 5%. And then our next best difference, which is essentially splitting our warm blood type horses from everything else, is only about 2.49%. And so there's very little amounts of variation in the genome that explain breed. And so at the moment, it's really a very coarse interpretation of what your animal's breed composition. Even with whole genome sequence data, it becomes very hard to predict when an animal is an intergrade. So that means if it's made up of more than one breed, because many horses share many of the same variants. And so some examples here, this horse 419, it is got a sort of thoroughbred and warm blood colors because it lined up best with those two breeds. And it was indeed a thoroughbred warm blood cross. So that one worked out really well. 851 here was a quarter, like very much a quarter horse, but it was starting to track closer to a thoroughbred and would be hard to identify as a quarter horse. So you can see it's not necessarily very straightforward just yet to be able to, at a very sort of accurate level, predict a breed based on the genetics. Some things in the future, I think the biggest thing in the future will be called pharmacogenomics, and that is how we can use what we know about a horse's genes for how it will respond to drugs or how it will respond to sedation. And so another good example of that we already know is our malignant hypothermia. So if we have that gene plus our trigger, then we have the disease, which is of high consequence. And that trigger is inhaled anesthetics. So if your horse would have, say, an orthopedic procedure and you knew that it had this gene, then you might have a different anesthetic strategy because of the potential for causing malignant hypothermia, or you might proactively treat it with a drug like dantrolene or something like that to make sure that we prevented this sort of pretty serious consequence. And so that's an example of where we can use genetics to inform therapy, particularly when it pertains to drugs. And there's some stuff that we're working on at the moment that hopefully will, go out into the wild world soon about different variants and how that affects sedation in horses. So the things that I'm going to leave you with here last, our biggest tip, make sure you get a good sample, and most of the horse samples that we are able to use are from hair. And then most important thing for that is that you need to include the roots, and we need at least 20 to 30 roots to be able to extract enough high quality DNA to do good quality testing. The last part is that even if you're not a breeder, it is still really valuable to test your horses, because that's a sort of citizen science thing, that if we know more about carrier state, we can do more to help know how these diseases are changing within our population. And also it would help inform you with some decision making. For example, if you had a quarter horse that had that malignant hypothermia gene, you would be able to tell your veterinarian to be careful with anesthetics. Another thing I would sort of recommend is that if you're considering genetic testing, it's a good idea to consult with your veterinarian. Not every veterinarian has, a background in genetics, but they will be able to reach out to someone that does, or some, institution like Cornell to help with those decision makings. That is all I have for now for genetics. I have two little announcements as part of my other job as sort of the equine programs committee here on campus and our sort of promotion of all things horses. One thing I wanted to make the people in this webinar aware of are our National Service Animal Eye Exam event that happens all through the month of May. So this includes large animals and our horses. You can go to this website here, our acvoeyeexam.org, to see if your horse qualifies. And then if you register in advance by calling this number, they're free eye exams through the month of May on Fridays. So I think that's something that would be really good to take part in if you have a service animal or assistance animal that is in need of an eye exam. The last part is that Giving Day at Cornell is coming up, which is on March 14th. And this year is the first year that we have a specific giving lane for equine programs here at the college and sort of wider throughout the campus. We're really interested in promoting everything equine. And so this is part of our launch for this. We have a really great mascot, Minnie. If anyone's been here for events here before, I'm sure they've probably met Minnie around the place. And with that, happy to take any questions.
Thank you very much, Dr. Donnelly. That's an excellent picture of Minnie, and I encourage anyone in the area to come to any event she's featured at. She's very sweet, and we do have some questions. The first one is regarding IMM, immune-mediated myositis, in horses. The person's wondering about a few things. One is triggers, if there are any known triggers beyond fevers, strep equi, and then also if you either have a horse that's been diagnosed or are concerned that they might have the gene, what precautions should you take?
Yeah, this is a really great question. And so it's what we would call a genetic risk factor, right? So not every animal that has the gene, and particularly not every animal that is homozygous for that gene will have immune-mediated myositis. And we've sort of changed. When it was first recognized, it was very much implicated in the immune phenotype, but we also know that this is also a reason for rhabdomyolysis without the immune component. However, about 40% of animals that do have the gene and develop the immune-mediated phenotype have had a history of strep equi. So it is a really big risk factor. In those horses too, vaccination can be a risk factor. And so this is something to take sort of pretty seriously when you're vaccinating your horses that have this trait. That it needs to be done sort of with supervision, and this is where we can use vaccines plus, say antibody testing, so we can see if our horses really need to be vaccinated yearly, or if we can sort of have a more tailored approach for the horses. It is a really devastating disease. I've seen plenty of cases out in California, and those horses tend to melt in front of you. We often can stabilize them, but very often they will recur, and so it's sort of a lifelong condition. And it seems to have a bit of a bimodal presentation. So we're able to see horses quite young, about weanling age. Or I've seen horses where their first episode has been like later teens. And, so it can kind of sneak up on you that even if you have not had a horse that's ever had a problem, you could have it later on. And I guess that's sort of emphasize the importance of testing for IMM. Particularly if you have a quarter horse.
Great plug for genetic testing, I agree. And there, we got a couple emailed questions and then a question this evening as well about, is there any, if you're talking about Wobbler's disease, is there any genetic testing available or anything on the horizon?
Good question. As far as I know, Wobblers, like a lot of things that we think about bone involvement, has probably a polygenic outside of the one known condition that results in a malformation of that area. So there's a disease in Arabians that causes what we call atlanto-occipital malformation syndrome. I've also seen that a very similar sort of thing in miniature horses. Probably it's also genetic, hasn't had necessarily the same workup as it has in Arabians, but in Arabian horses for sure we have a gene. We can test for that. Outside of that, in our say thoroughbreds and warm bloods, no, there's not necessarily a gene for that. And nor is there a genetic test for some of the other reasons for ataxia in those horses.
Right.
Yet. Watch this.
Yeah, get back to me in 5 years. And then you basically addressed this, but is there anything you need to know in terms of DNA hair testing, maybe determining reputable places to send genetic testing?
That's it. And I guess not being a paid representative of any genetic testing company, I'm not necessarily going to recommend one over the other. But what I would say is be very careful, because unlike our human colleagues where genetic tests are regulated by the FDA and where everything has to be sort of verified as causative, you can market an association test in horses without having sort of big data to back up that it really causes disease. And that's where so the PSSM1 story is kind of one where that has become a problem, where a PSSM2 test had been marketed to horse owners that was not validated, has a very similar name, so it became very confusing, and has subsequently, I checked recently, but should necessarily be taken off the market. So that would be my one sort of recommendation. Do your research, make sure you sort of talk with your veterinarian, and then sort of decide on what the most appropriate test, because not every test out there has been validated.
Good point. And this is, you touched on also a little bit, but maybe you could delve into it a bit more. I think you mentioned in regards to anesthesia, but someone's asking, how do genetic tests influence drug choice and dosing?
We're not quite there yet, but there is quite a lot of work. And the reason why there's quite a lot of work is because it likely will affect racing. So at least in our companion animals and in cattle, we know about genetic variants that change things like how fast an animal metabolizes a drug. And so this is, probably where some, trainers maybe run into a problem where they have like say the FEI said, you can't compete within this certain day based on what we know on horses. But then you compete your horse and you waited all those days, and then it swabs positive. And that horse might be a slow metabolizer. Now, there's definitely going to be a genetic basis to that. And there's a number of groups working on those. We don't necessarily have those tests out there just yet. But for sure, our group has worked on sedation phenotypes, and hopefully we'll have some information out there in the next year or so about sort of easily identifiable things that might influence the way that you sedate a horse. And then I think, really it will be sort of coming from the racing industry, because particularly drug metabolism is probably going to have a genetic test soon.
That's quite interesting.
Yeah.
And then seems like a more general question, but do you know if there are any if there's a genetic basis for either self-mutilation in horses, the stereotypy, or the white? I think they're just interested in the white lethal gene.
Yeah, so as far as self-mutilation, there is, at least in there's a repository of all known heritable diseases called the Online Mendelian Index for Animals, OMIA. It's run by the University of Sydney, and there is a registration in there for self-mutilation. I'm not sure if an actual causative gene has been identified, though, in that case. For lethal white, for sure, that is well recognized. So if you have a frame of error, that is a dominant trait, white, color patterning trait, that in homozygous form will result in a white foal that has a failure of the enteric nervous system to develop. So that one's definitely been worked out for a while, and is a well-known one to sort of, breed two frame of errors together, even though they're pretty.
Yeah. Yes, and I'm glad that's well known now, I think. Okay. And then there's also a couple requests to share the eye exam. So I think I'll just type that into the chat in case anyone wants to sign up. And actually, Dr. Nickelbein, who will probably be doing many of those, is also here tonight. And so could potentially answer questions about the logistics of that. But I think the best place to go is this website. Okay, I think we've addressed.
I'll just mention quickly, registration for this event opens in April, so it's not open yet, but you do need to pre-register. You can go ahead and call the clinic, though, to reserve your appointment, but you need to do both of those steps: register via the information on the website and also call the clinic.
Thank you very much. And so next month, I'm briefly forgetting who our speaker is. Do either of you remember? We have speakers through May lined up. April is on mud management.
Next month should be Dr. Fry, and
yes, it is.
be gastrointestinal
GI parasites. Yeah. And then we'll have in April of mud management on horse farms, and then May will be a unique one that'll be kind of a licensed veterinary technician's view of hospitalizing your horse at Cornell, and they'll give a lot of kind of insider tips on that. So that'll be fun. And thank you very much, Dr. Donnelly.
Thank
And we'll
you.
see everyone next month.